Hi, everyone. Welcome. Thank you so much for being here and for joining us today. This is our third installment of our Amp Up Your Devices webinar series, a series showing different examples of how our Fusion AX system can be used to study nanoelectronic devices in situ within the TEM. My name is Jen McConnell. I'm a product manager here at Protochips. And today we have a talented speaker who is breaching a very popular topic, which is focused ion beam sample preparation. FIB preparation can be challenging for traditional TEM studies, and you add another layer of nuance when it comes to in situ TEM, as proper connections need to be made from the sample to the support that don't interfere with in situ measurements, and so much more. And so the presentation today should shine some light on this topic for you, and you'll have a chance to ask questions as well. So a great opportunity if this has been a topic of interest for you recently. We do have 3 more webinars in this series that are studying electronic devices in situ with our Fusion system. The chat has the link to the webinar series, so please feel free to sign up. It's free, and this way you won't forget as they come up over the next few months, and you can share those with your colleagues if they have any interest as well. The more the merrier. So a couple of housekeeping items before I hand it over to the star of the show today. So everyone is muted due to a decent sized audience and to prevent accidental noise during the presentation. So if you have questions, and this is a great opportunity to ask some, there is a questions box in the GoToWebinar panel. Please put your questions in there, and then at the end of the webinar, we will ask our speaker and to answer them for you. And then we also have a survey that's going to pop up at the end of the webinar. And if you have 2 seconds to fill that out, we would greatly, greatly appreciate it. Okay, so that's enough of that. Let me introduce you to our speaker for today. So today we have Dr. Xiangli, or Dr. Shirley Zhong. She is the senior lead for FIB at the EM platform at the University of Manchester, which is one of Europe's largest electron microscopy facilities. And Shirley oversees the advancement of FIB techniques and the development of innovative and transformative solutions. She holds a bachelor's in engineering in precision metrological instruments, as well as a master's in engineering and a PhD in materials science. With a robust academic background, and coupled with the extensive experience in electron microscopy, she's developed a keen interest in the intersection of instrumentation and material science. So Shirley's dedicated not only to using electron microscopes for material analysis, but also to improving the quality and precision of material characterization. She's received prestigious awards from the Royal Microscopical Society and the Microscopy Society of America for her remarkable work in the field. And additionally, she dedicates her time to microscopy education and public engagement, fostering the development of future generations of microscopy and materials researchers. Shirley, thank you so much for joining us today. We're really looking forward to your presentation. It's a topic we get asked about a lot, and so I think this is going to be very well received, and you'll have several questions at the end. So thank you so much for being here, and take it off whenever you're ready.
Yeah, thank you, Jennifer. Thanks for the introduction. Hi, I'm Xiangli Zhong from the University of Manchester. I work as a cellular electron microscopy specialist, leading in the development of advanced FIB techniques. So today I would like to share with you our development on the FIB techniques for in situ TEM sample preparations. Apologize. Yeah, now go ahead.
Perfect. Yeah.
Yeah. So why we have this series of projects going on is because the companies like Protochips, they really developed very advanced MEMS-based TEM in situ cells, which open up the doors for the lots of in situ TEM characterizations, which in the past is impossible. But there is some challenges on the sample preparations too. That's why this project is to get in line with the rapid development of the advanced MEMS-based TEM in situ cells developed by companies like prototypes. So we have lots of prototypes holders and in-situ cells. So we are in the advantages of developing the techniques. And there are, as I said, there are many type of in-situ cells in our electron microscope center. And so we try to develop all the techniques for those holders. So today I'm just using some exemplar materials to show you how this development of FIB techniques can be used. These are only examples, but it can hopefully can used for your materials as well. And so in this, I will basically present two studies. One is in-situ liquid environment study, and also in-situ heating and irradiation study. However, we believe all these developed techniques can be used in a wide range of in-situ TEM bulk sample preparations, such as for liquid, in-situ liquid, gas, heating, electrical, and irradiation, whatever studies. We have developed 5 types of methods I'm going to present to you. The first development we carried out is for our liquid cell. At the time of development, there are almost no real in-situ liquid TM studies for bulk samples. I mean, using the chips, using this E-cells or MEMS-based E-chips for environmental study. The reason is because the sample preparation is really hard, because it's a bulk sample, if it's for bulk sample, and the liquid tends to scatter the electrons. Another challenge is because it's an E-cell, environmental cell, where the space is really confined. That's why at that time, most of the studies are limited to the nanoparticles or nanowires, where you just drop your sample on the grid, close up, that's it. But for bulk site-specific samples, it does need to get some way out to do our study. So in this study, we used the cold rolled type 304 stainless steel, and the chips we used are the protochips P200 liquid cell. And basically, we developed the hybrid method, which use electrochemical polishing with FIBing. The advantage of the electropolishing is it has large electron transparent area, whilst our FIB can have site-specific areas. That means whilst you have large electron transparent area, but we can pick up specific area which you are interested to do the in-situ liquid study. Of course, like I just now said, this is just an exemplar method or exemplar sample. In fact, for just say, for example, the electropolishing step, you can use something like mechanical polishing or ion beam polishing or ultramicrotone, which produce large areas, and then you combine with FIB. It's also feasible. So depends on your material. You can always alternate this method. So this is just a procedure. So what we do is first we use Thermopol 5 electropolisher. We then we use the electrolyte and we at minus 33 degrees. And however, the caution, or we found during the process, as we need to watch out the residual chemicals, if there's any residual chemicals or stress, especially during the cleaning. So this is very important during this step for electropolishing. So electrolyte, of course, can alter, depends on your material. And then we use SCM stamp mode and to observe or select the thin area of interest. Can see the setup, the left side photo is our setup with the stamp detectors, and the white bit in the center, the next photo shows the thin area. That's signal electron image with thin area appear as white due to the high generation rate of the signal electrons at the back, as the sample is thin enough. And the last two, the right-hand side two, is the stamp image for bright field and dark field image. Can see it's thin and can see the contours. So this is the area of interest. We developed the first method. We call it as cut and paste method, where you can choose an area of interest. So like the red square shows, then you mill it out, that area, and then you lift it up and place on the window. And then you use the PT to weld at the side. So sounds like not very difficult. In fact speaking, this is quite easy to do. So I hope you will find it quite accessible. But there are a few precautions or few points we need to consider. That of course you need to first choose your area of interest. Is that where you want to do, even for the broad ion beam polishing, electrode polishing, or whatever. So that's area you need to choose area of interest within that broad area, and then pick up the area of interest, use the FIB. And is it thin enough? So we use secondary electron or stamp image to make sure it's thin enough, especially for the liquid cell. The liquid itself scatter the electrons. So we need to make sure our sample is thin enough. And because of the chips, so when you tilt to 52 degrees, or other systems, because we use thermal Fisher's FIB in this study, which ion column to electron column is 52 degrees away. But other systems might be 54 or even 90 degrees. Anyhow, you need to tilt the sample during the process. So you need to consider the sample stability and the chip stability. So for us, we just simply make the sample stop with a groove, and then sort of like mechanically secure the chips not to falling off. For our experience, this method seems like it's quite stable, give us quite good results. During the large area cutting, you need to be careful that you don't want the so-called redeposition So you want to cut a large area because the redeposition might fall back into the window, silicon nitride window. That's why the large area milling hopefully can, especially for PT, because after the cutting, you need to do the PT deposition, and the deep PT deposition, it might fall back into the window area. That's why I try to make them as large as possible for the cutting. But for the FIB cutting, you need to optimize the speed and the cutting time. Higher current might be faster, however, it might produce more redeposition problems. And for the PT contamination issues, we try to use electron beam. However, the electron beam deposition for PT is slow. So there's another optimization similar to cutting speed. So you need to choose optimize between electron beam and ion beam deposition. Basically, you want to minimize the contaminations. This is the points, another points to consider. And of course, you need to consider if you will damage the E cell, because during the cutting, see when you place down your sample on the E-chips, you need to be careful that you don't like mill through the silicon base of the cell, because it's a liquid cell, you don't want to damage it. So there are a few points of consideration during the process. And like just now I said, we have the concern about the PET contaminations. So we developed another method, we call it glue method. Basically, similar to just now, you identify an area, mill it, and then you dip in the photo C, you dip your tip, your micro manipulator tip, no matter it's Omniprobe or Easy Lift or whatever, which companies micro manipulator tip, and dip into the glue. This we used so-called SCM glue, which can cool under the electron beam. So dip that glue and to from D, C and D, you dip the glue, and then at E, photo E, you can see the tip is tipped with glue, and then the glue, and then the tip basically touch with the area which we almost cut free, and then after it's filled in the glue and the glue attached with the sample, then we use high current to basically shining on the or use the electron beam to cool the area, the glue area that's between the tip and the sample area, and so it will cool. Once it's cooled, and then you can cut free of the sample. That's F, photo F, and then at photo G, you can see the samples lift up, and then you paste, you place the sample on the prototype, and then you cut free of your tip. So in this method, the glue method, basically we avoid to use the PT in order to avoid the PT contaminations. So it depends on the what is your applications. Say, for example, this method can use for electrical conductivity studies, however, because you have less PT contaminations. The third method, we call it force method, basically use the electrostatic or van der Waals force method. As the same thing, we use the electrochemical polishing, and you have a large area of a thin area, and then identifies some basic quite loose area, and just use the, when your tip approach the losing area, just some slight touch, and you can find some pieces can attach to your tip, and then you just place your tip on the window. And the advantage of this method basically is quite clean, almost no contaminations. But only disadvantage, of course, is say, for example, during our liquid, we cannot flow the liquid. Basically, it will be swept away. But for like gas experiment, in situ gas experiment is quite good. And really, so it depends on, I think, various applications, but this is another alternative for the in situ TM sample prep using FIB as localized method. And this is our results, just some to show the liquid environment observation, and can see the window, and the initial status is B, photo B, whereas in photo C, after 72 hours in the distilled water, can see the oxide formation, the square ones. So we also did the EDEX studies, so you can see the EDEX spectrum imaging, whereas ion can see the ion particles and oxide, ion oxide particles. So it's absence of chromium, and can see the spectrum there with high oxygen. So that's we actually observed from like 1 hour, 10 hour until 72 hours. So this is the 72 hours. So you can see how this oxidation formed in the water. And another project we've carried out is that we need to we need to use the high temperature cells for high temperature studies and for irradiation studies. Basically, we have two types of holders. In Manchester University, we have the e-chips from Protochip. I think you are all familiar with this, hopefully. And basically, you have the chips with holes where is the electron transparent area, and it can go up to the sample holder can go up to, sorry, not sample holder, the chips can go up to 1200 degrees C. And another type is the conventional sort of heating furnace type, Whereas also in Manchester, but in our neighborhood university called Huddersfield University, where they have the irradiation capability. That means irradiation under the high temperature, which can go to 110 degrees. They use the conventional, so because of this type of holder, so they use mono grade or carbon grade for high temperature. And in this exemplar sample, we use the in-situ heating of silicon carbide because it's quite important barrier materials for tracer particles in fusion industry. It provides structure support within the high temperature reactors, and it operates at 800 to 1010 degrees. At this high temperature, the platinum can corrode the silicon carbide. That's why we need to study the radiation resistance of silicon carbide under high temperature. And also the TM studies as to compare the room temperature and high temperature, what happened during the high temperature and irradiation. So in this study, we used the thermal e-chips from protochips, and it has a silicon carbide substrate with integrated electrical contacts. And for conventional FIB half grades, we used moly and copper. Copper is just for mechanical study, mechanism studies. Later I will explain this. And why we conventionally we use, or in the past, the universities use the Huddersfield's conventional holder. However, there's a problem that the PT react with the silicon carbide at high temperature. You can see the before heating and after heating. It's basically the PT oil diffused into the silicon carbide from other phases. And the reaction is so bad under about 1000 degrees C. So we have to work out a new way. This is a conventional method. Basically, you need, this is a conventional method. Basically, you lift out your sample and then placed on the grid, but there is PT contaminations. The new method we developed is to make use of combining in-situ lift out and redeposition. Basically, you mill your sample like our conventional in-situ lift out sample and thin it down, and then lift it out using in-situ lift out, which is an optical system where you have a glass needle, and when you approach the thin foil, the thin lamina, and you will lift up the sample, and then we place this onto our in-situ, onto our e-chips. And the protochips advantages, you have so many holes basically, and you can place lots of samples. So this is one of the example where we place the sample, and then instead of using PT to do the welding, we just draw the holes at the side. This way, it secure the sample and the prototypes. We did a few studies. Up to now, we have a few PhD students has using this way, and it's all successful. So it's quite, in our university, it's quite a matured technique now. You can see this is what we've done. We've done a few samples can be placed on the prototypes, and you just draw mill some holes and lines, and secure the sample and the surface. You can see the results quite encouraging. See under room temperature, can see the PD silicates, but after heating at 1100 degrees C and under about 1 hour, can see the carbonylation of silicon carbide and platinum contamination clusters at silicon carbide boundaries, so can see clearly. And the most important to the FIB development techniques, we found it's quite stable, and there's no reaction compared to the conventional PT welding. So this is quite encouraging. However, we want to know more about why it can work. So we tested another way, which is in principle the same or similar with our pivot just now. That method we call pivot, and this is side-to-side method. So basically, you lift up your sample like conventional TEM sample, and the key thing is you need to mill a plate on the holder and a grid holder, and then you draw the lines and mill the lines in a photo C. You can see the sample is welded to the welding is successful from the laminar to the monograde. This how we make it, and can see under the in situ irradiation of 600 EV xylene 1 hour at 1000 degrees C. The room temperature, there's no reaction, and then after irradiation at high temperature, can see irradiation induced voids. Sample is quite stable. Then we also did some comparison studies, the before the in situ experiment and after in situ experiment, because we want to know if there's any like moly or the redeposition material diffusion or reaction. We did EDX studies, and it proved as quite stable. So there's no diffusion, except we can see the coexistence of crystalline disordered regions and stacking faults due to the irradiation of 600 EV xylene. And another way of doing this side-to-side method, which avoid using the GIS, no matter it's PT or tungsten or carbon, is from welding your sample from the top. You can see the photo A. You have this like V-shape holder, and then you can mill a platinum flat surface, and then approach your sample and to the flat surface and mill lines along these two holder, the grid holder, and the sample, and then it basically collects welded. Under the electron beam, you can see different angle that it's all weld very well. It's quite stable as well. So for both side to side, just not a side to side method, or the top down, where you draw the lines, draw the line of holes from top of the sample to your prototype prototype window. We just want to understand how it's weld. So basically, we used silicon carbide as a exemplar, and approach to the mono grid, and then we basically did a series of line milling, and then left a thinness area, so that we can do a more detailed study and see what happened to the interfaces. And we can see that in the intercollection, or the welding point between, in this case is copper. We also did the mono grids for it's all similar, the results basically between the grids and silicon carbide, and the welding points are mixture of amorphous phase and crystalline and nanocrystalline phases. That is typical redeposition materials structure. We zoom in more on how it collects. See in area B, you can see the left side is silicon carbide, right side is moly. In fact, for example, for B1, photo B1, spectrum imaging B1, at moly side, you can see the silicon at the moly side, whereas B2, photo B2, you can see the moly signal on the silicon side, so that the redeposition material fall onto both sides, whereas when it's thick enough, so it's just joined together. So because the redeposition materials always has gallium inside, so from the gallium B3, you can see it's all filled both sides of the redeposition material. We can see it's basically from B4, it's mixture of amorphous and crystalline materials. And we zoom in more to the exact the collection point C, and you can see that in the area of collection points, it's basically mixture of silicon, gallium, and moly. And we do line scan. When we do the line scan throughout this redeposition material, basically you can see it's mixture of both materials, and it's go into, say, the moly has at the silicon side has silicon, silicon side has moly, and moly side has silicon. So that's the way it weld, do the welding. To me, is if some of you come across with the friction welding to me is quite similar to friction welding, although we don't have friction. It's like this is ion beam redeposition. However, like a mechanical friction welding, where you're not using conventional foreign materials to do the welding, but it's just friction of two materials or two welding parts, and then it's basically generates the same material and mix in between and do the welding. I mean, this mechanism is quite similar to that. So we try to zoom in and see what exactly is the mechanism. So it looks like it's basically the both sides are generating the redepositing materials which filled in the gap. And so this is the mechanism of, we do some drawings just to explain this. So yeah, during the milling, the gallium ions, when it bombard on the when you draw the line, it's basically draw the milling line. Basically, it's gallium ions bombard on the silicon carbide and moly or copper or whatever grades you use, and then this the ion matter interactions basically bombard out the atoms. Molecules, which most of the molecules will be of nanoparticles, will be sucked away by the high vacuum, but some of it will drop back in the vicinity of the milling area. So as those nanoparticles, those redeposition materials usually appear as nanoparticles. Those nanoparticles or atoms will agglomerate and tends to attach into the holes or voids or rough surfaces. That's why it quickly filled the two samples together. That's why no matter it's revert method where we put our sample on top of our prototypes, or is the conventional grid method, the principle is the same. Basically, it's a redeposition material filled or weld these two material together. So in these studies, basically we developed two methods. One is we call revert method for MEMS-based chips, and side-to-side method for conventional grades. For the revert method, you first mill your sample on your bulk, whatever localized area you wish to study. Just choose that area and mill the thin laminar, and then use the in-situ lift out optical glass ladle to lift out and place on the prototypes holder, the holes, or if you have the gas cells, or if you have electrical studies, you just place on the wherever you need, you are interested. Say, for example, if you are doing electrical studies, you can place it just next to the electrode, and then you just draw some lines or holes at the side, and then it will weld your sample. Whereas you no need to use PT, which can contaminate your sample. Another way is, of course, the side-to-side, we call it. You lift up your sample and place at the side or top of your grid and mill the lines, and then it will secure your samples, and the and a grid. So basically, these two methods we developed, we found, say for our sample is silicon carbide, where even the redeposition materials, they are just quite stable silicon and carbide, whereas PT is quite reactive, so it's really an advantage over PT. And it's quite clean, sort of. So we are quite happy and for this development. And yeah, our users, PhD students, are frequently use this. And so basically, for the series of studies, we developed the cut and paste method, where you use a broad method. You have a broad area of interest, no matter what method you use, ion beam or electropolishing or ultramicrotome, and then you identify the area of interest and lift it up and place or paste on the grid on the prototypes, and use the PT to weld it. Another method we call the force method is for using the electrostatic or van der Waals force, whereas it's quite clean. But the thing you need to watch out is you cannot like have a flow of liquid, maybe gas or electrostatics is okay. And the third method we developed is the glue method, where just use SCM glue. The glue curl under the electron beam. So, and the fourth method is the rivet method, where you can place your sample using the in-situ lift out and just draw some holes or lines on side of your sample, and then you weld between your sample and the grid and the chips from top down. And the fifth method developed is the side-to-side method, where your sample is the side of your grid. You just draw lines, then it will weld together. So basically, we proved that the hybrid electro FIB sample preparation, I mentioned the first three as is quite good for environmental TM dynamic observations. And the in-situ liquid cell studies we used in the first three methods, we can observe the formation of the oxide with time in water. And the rivet method under high temperature observations is, and of course the side-to-side has been very successful. And we also studied the mechanism of redeposition welding, because we want to understand why. And now we know the answer, and so we tried to optimize those methods. Now it's quite successful, quite a popular method in our center. And in fact, the final conclusion is that the principle of the developed series of advanced FIB techniques for TEM in-situ studies can be used for other many types of in-situ TEM studies, including but not limited to in-situ electrical studies, gas environmental studies, or whatever. Maybe in the future, some more protochips may develop some more MEMS-based cells, we need to catch up with protochips, basically. Thanks for protochips. Very good job done. So we're trying to catch up with them for the new technique development and open up more opportunities for materials research, and also I think the biomaterials research as well. Yeah, so this is my brief introduction or presentation to our work recently, focusing on the in-situ TEM sample preparation. Any questions or discussions, please feel free to ask. Thank you.
Thank you so much, Dr. Zhong. That was a really great presentation, and as expected, we do have several questions related to some of the techniques that you've shown. I think the audience really appreciated seeing some new FIB preparation techniques. It's always a discussion between us and several different researchers. So I will go through the order that we received them, and if we run out of time, I will definitely connect you with anybody we don't get to, and maybe they have some additional information you can give them.
Okay.
So for the first question here, it says, for the second method with the glue on the tip, how much time do you have to move the manipulator around and touch the region of interest before the glue solidifies? Can you provide a little more detail about what to expect when using the glue?
Oh, and the glue, in my opinion, you can use very high current, because we tried various parameters, and then I realized you might need to use if your SCM has available of more than 10 nanoamps, you would tend to use higher current, then it will cool faster. I don't know the chemistry behind, but it's quite quick, basically. I think maybe 1 minute to 3 minutes, usually. And then I just cut it free, and then I realized it's quite stable. So right now, basically, in our lab, we try to use 20 nanoamp for a FIB SAM, FIB E column, 20 to 30 nanoamp, and just for 1 to 3 minutes, it just cools, and then you can just securely cut free of your sample. It doesn't take lots of time.
Great, thank you so much. The next question is related to 2D materials, and they're asking, would it be sufficient to just transfer the 2D material onto a chip to make contact with electrodes? So it sounds like they're asking if like the van der Waals method would be sufficient for making electrical contact for a 2D material. What do you think?
Yeah, I think it's feasible. I think yes. You can approach your needle to the 2D material, say nanowires or whatever, and put it over there, and then just retract. But sometimes the thin foil or 2D materials may come back with, we may stick on your needle and it won't go away. Then you need to try again. That means you place say for example nanowire back again, and then you lift up, and then again you may have a few tries until it stabilizes, until it really drop on the surface. It's possible. And I've tried, and see sometimes it doesn't really want to go where you go. It doesn't really listen to you. It just do whatever it want to do. It just place somewhere else. I tried to use at the very early days. I tried to use ion beam see, just mill it off, but actually it doesn't work. So basically just a few tries to put it down. If it go back and just put it down again. Yeah.
Do you feel, and actually, I'll follow up that question because I think it might answer another question or two from some others. Do you feel like that method then makes good electrical contact to do like electrical studies with the electrodes as opposed to using like platinum and such to make the connections?
Yes, I think to me, I would, if it's for the electrical, see in the glue method, actually the last photo, in that study, actually we used electrochemical cells, which has the electrodes on the chips, on the proto chips. So after use the glue method, we indeed use electron beam deposition, a thin layer of PT on the between. Maybe I can, if you can still see that photo. Let me see if I can find that one. Oops. Yeah.
Yeah, there were a few questions related to the glue method. How can you ensure that there's good electrical contact with the platinum electrode?
Yeah,
And
you see the eye,
yep.
the photo eye. Yeah, can see there's a little bit electrical contact. We use the E-beam. We use E-beam because E-beam has this is a thick SEM, so the E-beam slower, but the spread is not as bad. So we use the E-beam deposition. You can see the electrodes. Actually, we indeed use the PT, electron beam induced deposition, to do some contact. The good thing about this method is really you don't have the PT contaminations where it can affect your conductivities or whatever. Yeah.
Yeah, definitely. Okay, so the glue stabilizes everything, and then you make the electrical contact with the E-beam then instead. Gotcha.
Yeah.
Okay. Let's see here. And so this question was also sort of related to it. And I think it may have been answered, but maybe there's a little more detail you want to give. They said, regarding the glue and the van der Waals methods, the lamella deposited on the silicon nitride membrane seems fixed after being laid down. So then why wouldn't this method be applied to lamella for that were extracted for electrochemically thin samples? So why? I think they're saying if you get a good connection with van der Waals and the glue method on silicon nitride, why would those not be sufficient by themselves for doing electrical connections for electrochemistry? And it sounds like you're saying they're good for stabilizing, but then you still need something to make a good electrical contact to the electrodes. Is that right?
Yeah. You need to collect, is it, between your sample and the electron. You need to use some conductive materials to collect them, to make the contact with the electrons.
Okay, great. We have a few more. Let's see what we can get through here in the next couple of minutes. Dr. Zhong, thank you for your helpful talk. I wanted to ask, which pattern should I use for welding without GIS, line, RCS, or CCS? Are there any differences?
So for PT, so that means the question is for PT deposition, the GIS. I usually use the code Situ TEM. So it's basically I don't use CCS. The CCS is like so Situ TEM is more uniform. I should say that way, because you deposit in a way that sweat everywhere. But it's if you use CCS, that means you are doing it line by line, It's not as uniform. So the Situ TEM go up and down, left and right. That's the best. Yeah. I hope this answered the question.
Great. I'll let you know if they follow up with anything additional.
Yeah, if anything.
Yeah. Last couple I think might be a little bit quick. So, hi, I have a question regarding the rivet method. How do you exactly make the sample stay attached to the chip?
So sorry, which method?
Rivet, the rivet method.
Yeah.
They're curious how is it exactly staying attached to the chip?
Yeah. It's this method. Yeah. It's just you draw the lines or the like the red arrows. So basically the sample, see if it's silicon carbide or it's silicon, and then if your the protochips is made of silicon carbide. So this basically your chips material and your sample material, because you wear through both of them, you mill through both of them. In between them, there's some redeposition materials. And then the redeposition materials, they basically mix and redeposit at the gap area. So it's how it weld. That's exactly the mechanism studies. It's like this. Yeah, it's like this. So between grid and sample, say silicon carbide is your sample, say for example, and the copper grid or protochips, silicon nitride or silicon carbide, or whatever. So in between these two, you have the redeposition material which weld together. So-called weld is filled with the redeposition materials. If I answer that question.
Perfect. Yeah, I think so. And here's the last one. And so they're still here, I see as well. So we'll go a couple minutes over. Hello, Professor. Thank you for your wonderful introduction. The question is for your rivet method. What is the thickness of your lamella on the grid window? And could you please mention more how to thin the lamella before transfer to the window?
So two questions. One is how thick is my lamella and how do I thin the lamella? Am I correct? Yeah.
Before transfer, yes, that's right.
Yes. So that yeah, this. So basically, the typical lamella, the thick lamella, is about 2 microns, 1 to 2 microns. Usually we do 2 microns, and then you thin it until electron transparent. That's under the STM. If you can see the white, means the electrons are the shoot through the thin foil, which generates more secondary electrons, which make the image as bright. And then you cut free of this lamella, and then you lift up. Once you lift up, and then just placed on the sample, on the protochips. You just do some milling rivet method. Just do some rivet and just like a mechanical rivet, like a bolt. Then it will secure it. So you basically you thin it first, and then you place on it and do some milling at the side, and it will secure the sample.
Perfect. We made it through all the questions. And I'm not sure if you had your email address on the last slide or anything, but if anyone has questions, if it's okay to reach out to you, we can certainly connect them with you if someone asks another afterwards. Dr. Zhong, thank you so much for putting this together. Really informative. And I think the audience seemed to really enjoy it. So thank you very, very much. And thanks everyone for coming today. If you don't mind filling out the survey before you leave, that would be amazing. Dr. Zhong, have a great afternoon and a great weekend, and Happy New Year.
Thank you. Happy New Year.
Thanks. Bye. See you later.
Bye. See you later.